Today we're going to look at a real-world example of how leaf structure connects directly to plant survival. This case involves a common plant you see almost every day, grass, and how it responds to heat and drought stress. Alexandria noticed that during an extended period of high temperatures and low rainfall, her grass blades began to curl inward toward the midrib and looked rolled or closed. After a heavy rain, the blades returned to their normal open shape. This tells us the change was rapid, reversible, and tied directly to water availability. So the question becomes, what structures in the leaf allow this to happen? And why would this be beneficial? Pause the video and write your response to reflection question 1 in your lab notebook. Think back to the external leaf features you studied earlier. Ask yourself, does grass have broad leaves or long, narrow leaves? What type of venation do grasses show? How do the veins run from base to tip? Those questions should point you toward the correct classification. When you resume, we'll move on to the next question. Focus on the internal structure of monocot leaves. Recall, we discussed large, thin-walled cells near the upper epidermis in some grasses. These cells lose turgor pressure when water is scarce. When they shrink, the leaf folds or rolls. Think about which cells were associated with leaf rolling, drought response, and changes in cell volume rather than permanent tissue damage. Those are the cells you were looking for. Now connect structure to function. Pause and write your response to reflection question 3. Ask yourself, what problem does heat and drought create for leaves? What happens to water loss when a leaf surface is fully exposed? How would rolling the leaf change the surface area exposed to air and sunlight? Also consider stomata location, transpiration rates, and how reducing exposed surface can slow water loss. This response is not damage, it's a protective, reversible physiological adaptation that helps the plant conserve water until conditions improve. This case ties together everything you've learned about monocot leaf structure, specialized epidermal cells, structure-function relationships, and environmental stress responses. Grass isn't just wilting. It is actively responding using specialized leaf anatomy to survive drought and heat, then reopening when water becomes available again. In this lab, you examined how leaf structure supports function across many plant types and environments. You compared monocot and eudicot leaves, identifying differences in venation, shape, and attachment. You then studied internal anatomy, locating the epidermis, cuticle, mesophyll, and vascular tissues, and connecting palisade mesophyll with photosynthesis and spongy mesophyll with gas exchange. You explored how leaves adapt to environmental conditions by comparing mesophytes, xerophytes, and hydrophytes, seeing how the tissue organization changes to conserve water or enhance gas exchange and buoyancy. You also examined specialized leaves modified for support, protection, storage, and nutrient acquisition, including tendrils, spines, succulents, and carnivorous traps. Across all these examples, the same basic leaf tissues were present, but arranged differently to meet different challenges, reinforcing the key idea that structure follows function and function reflects the environment.